Alright, here's our video for tomorrow. Sorry about the delay in getting this video up. Just ran out of steam yesterday and had to shut it down. So this chapter 7 is an interesting one. Is that it takes three biological processes: developmental biology, ontogenesis the development of cancer, and what is the third? I can't think of what the third is right at this moment, and walks you through the genetic issues that have to be overcome in order to accomplish those processes. And so this video is going to cover the developmental aspect of that. On Wednesday, we're going to focus our class time on talking about ethical issues in genetics and how do we build a robust ethical foundation to answer really difficult questions through. And then we'll also focus in on the oncogenesis and the other aspect of biology as well. All right, so some key terms with regards to developmental genetics. Oh, gosh, these are coming out of order. Let me do this. Oh my goodness, that's really frustrating. So cell differentiation is the process in which a cell moves from being totipotent in the zygote all the way to being unipotent or monopotent, different terms for the same thing. And basically you inactivate some genes and activate others, and you're going through epigenetic changes that permanently alter the cells. Oftentimes this process is cued not by intrinsic factors, but by extrinsic factors. Most of those are going to be cell-to-cell communication or cell adhesion to an extracellular matrix or other cells that are going to cue the cell into transcribing certain genes that are going to move it through those stages from totipotency to unipotency. Now, morphogens are chemicals that are present during the development, early stages of development of an organism, and basically set up a concentration gradient. And the concentration gradient of these soluble materials that are in the extracellular fluid trigger the transcriptional change of the cells. And so when they are in high concentration, they tend to signal a certain set of genes to be transcribed, and when they are in low concentration, they signal a different set of genes to be transcribed. And a great deal of the development in animals is the result of these concentration gradients of morphogens. And we'll talk about one of these specifically in a little bit. Homeobox genes or master control genes. These are genes that set up the axes, the major axes of the body, the anterior, posterior axis, dorsal, ventral axis, left and right axes, superior and inferior if you've got a upright walking organism. And these genes all code for transcription factors, and this is why we call them master control genes, because if they code for a transcription factor, they are going to alter the expression of other genes. And so between morphogens and homeobox genes, you explain the bulk of the genetic aspects of development in organisms. SRY gene, we've talked about this often. There's a region surrounding the gene, and then there's the main gene product of this area that basically is transcribed and then translated into a protein that results in most of the male sex differentiation, and we'll talk more about this process in a few slides. So R-spondin 1 is a gene that actually finishes the female sex differentiation process in mammals. And so in mammals, at least, just the absence of the SRY gene is not enough to get a female that will become sexually mature and will function normally, you also need to have the expression of R-spondin 1. R-spondin 1 will then lead to an upregulation of certain elements that will finish the process of developing a fully functional female mammal. Last key term, induced pluripotent cells. We'll talk about this a great deal in our class time on Wednesday, but it's basically they are cells that are reprogrammed from an adult to believe that they are embryonic stem cells. They're that pluripotency where they can develop into basically any cell type is restored, and this reprogramming is really nice because now you have a cell that is pluripotent, can really develop into any cell type in the body, but it also came from your own cells, so it's not going to read foreign to any of your immune system, any of the components of your immune system. All right, so the most common or the most talked about, the most studied of our morphogens is called sonic hedgehog. And so here you can see the gradient. Let me get my pen. Haven't used this pen in a while. I feel it's going to feel nice. So here's our gradient. Here would be a really high concentration of sonic hedgehog, and here would be a very low concentration of sonic hedgehog. And one of the most well understood implications of that of the gradient of sonic hedgehog is in the neural tube. And so in the neural tube, you get basically from this would be typically the dorsal side, and then here's the ventral side here. And so in where there is a high concentration of sonic hedgehog, you develop some motor neurons. You can see that right there. And then some other interneurons that are going to become sensory neurons. And then when there's a low concentration, you develop all sensory neurons. And so what this is going to do is basically tell cells that are already going to develop into neurons and glial cells, basically to develop either into motor neurons or develop into sensory neurons. And all of that is basically controlled by the difference in concentration of this morphogen, sonic hedgehog. And so this is something that can be altered either purposefully or accidentally because of some developmental abnormalities, and then you can get a malformed and unusually formed neural tube, which will then develop into a malformed nervous system that would likely lead to the death of the individual. All right, so other than morphogens, Hox genes, and even Hox genes may be the most important gene or the most important element controlling the development of an organism. And again, these are genes for transcription factors, and these are genes that set up the major axes of the body. So here's showing basically the anterior posterior axis, here also anterior posterior, although since humans are upright walking, it would be superior inferior. And you can see that there's an enormous amount of similarity between the Hox family, at least Hox C. So there are 4 Hox families: Hox A, Hox B, Hox C, Hox D, and these are all gene families. And there's an enormous amount of similarity between the Hox genes found in Drosophila and the Hox families found in humans. Now, we have more Hox genes families than what you find in Drosophila, but the reason for the enormous similarities is, again, keeping in mind that these are major control genes. These are coding for transcription factors that are going to establish the anterior posterior axis. And so regardless of whether the organisms share ancestry or not, you would predict that there would be an enormous amount of homology between the two organisms, and it's just a matter of how do you explain that homology. Is that homology from common ancestry, or is that homology from a common design? But regardless, you need the programming to be very similar because this is establishing the major body axes. And so for any bilaterally symmetrical animal, you would predict that there's going to be a great deal of similarity in these Hox genes. And again, these Hox genes work along with morphogens to basically signal extrinsically to a cell what genes it needs to transcribe, what genes it needs to, methylate and shut down permanently. And so a lot of the relationship between Hox genes and morphogens were determined experimentally. The textbook talks about research done on chick limbs, and both Hox genes and morphogens work together to determine the distal and proximal end of the limb, so that at the distal end of the limb you develop digits, and at the proximal end of the limb you do not develop digits, but you develop the portion of the limb that is going to articulate well with the other components of the joint at that limb. And so both Hox genes and morphogens work together, and any errors in either basically resulted in the same abnormality. That is, the inability to establish proximal distal end and the inability to develop digits properly. All right, so how do mammals become male? And so here we've got the SRY gene, and notice there's something really important illustrated here, that there are other factors involved. As should be predicted, the SRY gene does have modifier genes, and interestingly enough, some of those modifier genes are on the X chromosome. And so, but ultimately, if SRY gene is transcribed and then translated into SRY protein, you are going to start the process of developing a male mammal. So once this SRY protein is translated, it will convert an undifferentiated gonad into differentiated testes made up of both Sertoli cells and Leydig cells. Sertoli cells are the cells that in a sexually mature male are going to take part in gametogenesis and ensure that germ cells develop into spermatids and not into oocytes. But early on in development, these Sertoli cells express a gene that codes for anti-Müllerian hormone. And so as long as you have the receptor on the proper cells, this inhibits the Müllerian duct, which would develop into the female reproductive tract. And so that leads to the degradation of that material and the resorption of that material. And then the Leydig cells, these secrete testosterone. And once testosterone starts to show up, it triggers the Wolffian duct to develop into the male, a complete male reproductive tract, complete with the male genitalia. Testosterone also works with brain development to develop the psychological features of maleness, and also is converted into dihydrotestosterone, which will then lead to the development of male secondary sex characteristics, the growth of body hair, the growth of facial hair, and characteristics of male humans, although there are secondary sex or male sex characteristics in most mammals. Think about the one that came to my mind was a lion, the development of that really large body size and development of the mane, or male silverback gorillas, the development of that really large size and that silver, the silver hair that develops on the back. And so all of this basically is triggered by the transcription and translation of the SRY gene. And so While this, even just having this SRY piece is enough to develop maleness, it can be inhibited by other factors. You notice anywhere where we see plus receptor is a place where this process could be inhibited if you've got a mutation in the gene that codes for that receptor. Now, something that's important to note is this SRY gene is pretty close on the chromosome, on the Y chromosome, to the PAR1 region. And so you in the event that you get an uneven crossing over, you can actually transfer the SRY gene to the X chromosome and create what we call a pseudo Y chromosome. And again, the presence of that SRY gene is typically enough to determine and to develop a male individual. Now, then you would essentially result, if you have two X chromosomes, but one of them is a pseudo Y chromosome, you would essentially have an alternative way of having Klinefelter syndrome where you have two X chromosomes plus a Y. Even though you only have a portion of Y, you have the portion of Y that determines maleness in mammals. And so you would essentially develop Klinefelter syndrome where you would have a male individual, but they would likely be sterile. All right, so what are primary issues in sex determination? What are the most common issues? One, mutation in the SRY gene, or rarely a mutation in SPO1 that R-spondin 1 that determines female, that results in a fully formed female mammal. Those are probably the most common disorders, that should say on, or disorders in the steroid biosynthesis. That's nowhere even close to in. I don't know what happened there. Disorders in the steroid biosynthesis, so disorders in some of the proteins that are necessary to make testosterone or make estradiol or some of the other estrogens. Mutations in the androgen receptors, so again, mutations in those genes that code for the receptors that receive or bind those sex hormones and then trigger a transduction pathway that is going to result in cellular differences. Defects in the AMH gene, this is anti-Müllerian hormone. This is the gene that again codes for that anti-Müllerian hormone that will result in the inhibition of the formation of the female reproductive tract and ultimately the resorption of that. XO-XY mosaicism. So if you have a mitotic non-disjunction event in a male individual, some of the cells could end up getting no sex chromosome. And so you could end up with a situation in which some of the cells only have an X chromosome and not a Y, and then the other cells have the normal XY condition. And the result of that is basically you've got some cells that are not male and some cells that are male in the same individual, and will oftentimes lead to issues in the development of some of the the male secondary sexual characteristics. Mutations in modifier genes, I mentioned that in the SRY gene specifically, that there are modifier genes that regulate its transcription, and some of those genes are on the X chromosome, but some of those genes are also other places on autosomal chromosomes, and any mutations in those modifier genes can alter the expression of the SRY gene, and then create some issues in developing all of the characteristics associated with male mammals. All right, last thing we need to talk about, and this we're going to spend a good time on in class tomorrow addressing this, is stem cells. So basically the progression through differentiation is all controlled genetically, although the genetics are influenced by extrinsic factors. But basically we move from a totipotent cell in the zygote, wow, that should say zygote, the G is missing. You go from there to a pluripotent cell in the blastocyst. So the blastocyst, we haven't really developed the germ layers yet, and so all of those cells are pluripotent. Once you get to the gastrula stage and you've developed the germ layers, ectoderm, endoderm, and mesoderm, you have very few, if any, pluripotent cells left, and they're basically at that point we call them multipotent cells, and they can typically become any cell in that germ layer. And then from there, ultimately, you get to unipotent or monopotent cells in the adult, where they are capable of basically becoming a single cell type. Now, there's some exceptions to this. There are some stem cells still, like hematopoietic stem cells, that can develop into any of the blood cells. There are some stem cells in your skin that can develop into any type of epithelial cell, and even some cells associated with other tissue types, limited. And so there's obviously an enormous value in pluripotent cells that are capable of basically developing into any cell type. But we've got some issues of, well, where can we get them? And we find them in the blastocyst stage of development. So this is a very early embryo. And you obviously can't do that non-invasively, so we've got some really big ethical concerns associated with accessing these pluripotent cells. Questions such as, is it ethically acceptable to form embryos in vitro just for the purpose of accessing these pluripotent stem cells? Or is it ethical to use embryos that are, quote, left over after in vitro fertilization that might be discarded anyways? And so these are some questions that we're going to spend time on Wednesday talking about. But before we do that, we're going to work on developing a really cohesive and solid ethical foundation. Now, some of this is becoming not unnecessary to talk about, but we're getting pretty good at inducing pluripotency in adult cells. And so we're getting pretty good at taking cells from an adult and then reprogramming them into becoming what we call an induced pluripotent cell, or an induced pluripotent stem cell. And when we initially did these, that process of reprogramming also altered the some of our oncogenes and led to overexpression of proto-oncogenes and the development of oncogenes. And but now we're getting pretty good at this, that we can reprogram cells without leading to uncontrolled cell growth, the inability to control the cell cycle. And so we have an effective way to get pluripotent cells, and because they can come from your own body, it eliminates a lot of the issues with rejection. And so although there's still the question of, okay, well, what do we do with, again, these, quote, leftover embryos from cycles of in vitro fertilization. So we're still going to talk through some of these, and you can probably imagine this. There's still a strong desire to use embryonic stem cells. So as you prepare for class on Wednesday, think through some of these issues and be prepared to talk about some other biological processes and the genetic roles in those.